Hello everyone, in this video I will be discussing the metabolism of fats in the human body. So I think it's a good idea to start with a definition of metabolism. This is the one that I got from the textbook. So metabolism refers to the chemical processes occurring in an organism that are necessary for the maintenance of life. There are three main types of chemical processes. We are only looking at two in VC chemistry. The first one is the breakdown of food molecules into raw materials. So this is the catabolic reactions that I'm sure you've heard of in biology or health science. Breaking down of materials, catabolic, and the raw materials, once broken down, will be released into the bloodstream to be absorbed by body cells. The second process that we're going to look at is how that raw materials actually get used by body cells. For the most part, it's going to be via an anabolic reaction. So the raw material is used to rebuild the cell proteins or lipids or glycogen, if you will. But there's also a possibility that the raw material can be used for cellular respiration in the case of glucose. So it's not always an anabolic reaction. Now, there is actually a third chemical process that's involved in metabolism, which is the removal of waste, but we don't actually look at that in this course. But just keep in mind that that's the third main chemical process that you need to maintain life, if you will. We're going to start with the first part, which is the breakdown of food, which is done via digestion. So this is just a quick overview of digestion and the digestive tract, which you don't really have to write this down. But I'm sure it's common knowledge that there are four main areas in the body where digestion occur. You can argue there's really three, and the fourth one just to reabsorb water. The first area is the mouth, and this is where the breakdown of food begins. So there's a lot of chewing, if you will, and that increases the surface area available for digestion later. And there's an enzyme that's available in saliva that can react with certain components of food, namely carbohydrates. So carbohydrates start to get digested in the mouth. And then the second area is the stomach. This is when food is broken down further by the combined reaction of hydrochloric acid and some enzymes. So carbohydrates start to get digested, well, continue to be digested, and proteins start to get digested in the stomach. Small intestine, again, you have a mixture of enzymes made from the pancreas. The pH of the small intestine is actually quite basic. I think it's around 9. So this is when carbohydrates get digested further, proteins get digested further. And the digestion generally is complete in the small intestine, and then you will have small molecules being absorbed into the blood and transported around the body. Now, in the case of lipids actually start its digestion in the small intestine. So it's probably the molecule that gets digested the latest if you talk about location along the digestive tract. And so lipid doesn't get digested until it enters the small intestine. And the digestion of lipid generally involves two processes. The first one is emulsification. So the lipid molecules will get emulsified in the small intestine. And the second process is hydrolysis, which is the actual digestion or breakdown of triglyceride in this case. So emulsification and hydrolysis. Now there is a fourth component or area of the digestive tract, which is the large intestine. Usually by this point, you just try to reabsorb any water that might have been lost. Some of the fiber in the food may be digested by bacteria in the colon, but it's pretty much the end of the journey, and then anyways it's going to get excreted from the body. We'll talk a little bit more about the large intestine later when we look at carbohydrates, but for now we're just going to focus on the small intestine, which is where all the actions happen concerning lipids. All right, so let's start with emulsification, which is the first thing that will happen to a lipid molecule when it enters the small intestine. I think some of you probably have heard of this term before, emulsification. If you emulsify something, you're basically trying to mix, for the most part, lipid and water together. So mix lipid and water. Now, obviously, we know that lipid and water don't mix, but the whole idea of emulsification is you're trying to mix them in a way that you get a semi-stable mixture to form a semi-stable mixture. So it's not completely stable. If you spin it enough time, then the lipid and the water will split into two layers again. But if you mix it well enough, and stop at the right time before you overmix, then you get a semi-stable mixture. So examples of emulsification, say for example, if anyone has ever made mayonnaise at home, that's emulsification. So you mix anything water-based with oil, that's emulsification. Now, in for every emulsification process, you really do need an agent that can really help binding the lipid and the water together. In the case of lipid in the digestive tract, the lipid is emulsified using a molecule called bile. So bile is a hybrid molecule that has a hydrophilic part and hydrophobic part. It is made in the liver and stored in the gallbladder. So you can think of bile as like this little thing, if you will. So there is a hydrophilic. I cannot believe there will be a day when I say hydrophilic. So you know, if you are a purist, you can say polar. So it has a polar part, and then it has a hydrophobic or non-polar part. So it's a hybrid molecule, hydrophobic or non-polar. Okay. Well, of course I would do that. You know what? This is I do need this. No, I think we can just survive with that. All right. So in the small intestine, what happened with emulsification is that lipid is mixed with and emulsified by bile. This process would break down the lipid into smaller droplets. So the idea here is that the hydrophobic part of bile is going to surround the oil or lipid drops. I think they actually make this bit red, so I just do that. So what happens is if you have this blob of lipid, so if you have this blob of lipid, hold on, let me check. Yep. If you have this blob of lipid, the hydrophobic part or the non-polar part of the bile is going to surround it. So the hydrophobic part comes together and surround the oil, whereas the hydrophilic part or the polar part is going to be on the outside. So this is how it works. So you start with this huge fat globule, and once it's mixed with the bile, you end up with smaller fat droplets that is surrounded by bile. Now, this is the exact same mechanism that detergent works. So if you want to see how emulsification works or how it may work in the human body, you can just get a little bit of cooking oil and mix it with like detergent or this soap, and you should see the emulsification process and see how the large chunk of fat oil gets broken down to smaller drops. Okay, so the fat that we consume in food has to be emulsified first, and after emulsification happened, the actual breakdown of fat is going to happen, and via a process called hydrolysis. If you recall, the only fat or lipid that we look at in BCE is triglyceride. So it's a triester that's made of three fatty acid combining with a glycerol. So if you want to break it, you essentially under having the molecule undergoing hydrolysis to break down the ester link. The small lipid droplets are hydrolyzed. I don't know why I say not. Hydrolyzed by enzyme lipase. So every food molecule is going to have a dedicated enzyme that catalyzes the chemical reaction for its breakdown. So it's hydrolyzed by enzyme lipase into glycerol and fatty acid. So from here, you're going to have this happening. So the triglyceride is going to turn into glucose and fatty acids, which is the actual hydrolysis. And the reason why the emulsification step has to happen before the actual hydrolysis is because lipase is the water-soluble enzyme, and it can only interact with the surface of the lipid. So if I were to just have lipase trying to dissolving or hydrolyzing this really big fat globule, it would be very difficult because it could only interact with the surface of the fat molecule since it's water soluble. So the emulsification happens to break down the fat into smaller droplets, which would increase the surface area available for the reaction to occur. So you're trying to speed up the digestion process. In this case, I mean, you really have to, because the small intestine is literally one of the last stop in the digestive tract. So if you don't finish digesting the fat here, you're not going to be able to digest it at all. So the digestive tract really needs to try to speed up the process. So emulsification is important so that you could increase the surface area available for lipase to digest the lipid. Okay, so that's hydrolysis. Now, by this point, you now have completely digested the fat. There's only four components in triglyceride. So when you hydrolyze it, you just get four molecules being produced. What happened is, what would the body do with these molecules after it has been broken down, well, after it has been produced from digestion? And usually what happened next is condensation reaction. So this is no longer the digestion of fat. This is what happened to the fat once it has been digested. So after digestion, the glycerol and fatty acid would then be released into the bloodstream to be used by the body cells. So this is end of digestion, and these are gonna go into bloodstream. Okay. These parts, most of the time, what will happen that they will undergo condensation. Probably not to reform the exact molecule that was digested, but the body is going to use parts of it. So maybe a glycerol here, a fatty acid there, to reform, to make triglycerides for the body. So for example, you want to make your lipid in cell membrane or steroids and things like that. Obviously, this condensation reaction is going to require other enzymes and also water, because actually, I don't know why you need water. I don't know why I write water here. Water is going to be produced. The lipids will be stored in adipose tissue as a form of energy storage. So you break it apart and then you reform a different triglyceride, I suppose, but you form another triglyceride. But this is your body's triglycerides now. Hence the saying, you are what you eat. I mean, you do break down your food and then use the building blocks to make your own molecules. Right, so that's essentially the metabolism of fat. So you start with the small intestine. It goes from drops of oil, well, a globule of fat or oil, undergoing emulsification via bile to turn into smaller droplets. The droplets will then undergo hydrolysis to turn into glycerol and fatty acids. And this is going to go into the bloodstream where it undergoes condensation reaction to make body's fat, which, I mean, it has other function as well. It's not always energy storage, but energy storage is the main function that we're going to talk about in VC chemistry. I'm sure the bio people can tell you all about cell membrane. But that's the end of this drop point. Thank you.